Well, good morning. Thank you for coming. This presentation stemmed from a review of a book by Stephen C. Meyer called Darwin's Doubt. And Dr. Meyer has been in the news because he is a fellow at the Discovery Institute. And the Discovery Institute essentially describes itself as a conservative think tank. They produce what, in my view, is some very thoughtful, very good materials. I'm personally a George Gilder fan, so George Gilder is at the Discovery Institute, and if you're associated with George Gilder, you can't be all bad. However, the Discovery Institute has been a bastion for a movement called intelligent design. And the foremost promoter of intelligent design is this gentleman, Stephen Myers. I've met Stephen Myers 2 or 3 times. He is, without a doubt, a very brilliant man. He's without a doubt, too, a very godly man. He is a fine father. He's a fine husband. He's a fine scholar, but I must register my very profound disagreement with him. I could agree with his conclusions, but I don't agree with the way he got there. Let's put it that way. So what does his book argue? His book argues that this phenomenon in Earth history called the Cambrian Explosion cannot be explained by the contemporary theory of evolution. His reasons are manifold, but if I were to put them in the Reader's Digest version, it would go something like this. The Cambrian Explosion occurred over a, geologically speaking, short period of time, far too short for evolutionary processes to produce the vast array of animal diversity that we see in the fossil record. Secondly, if you're going to make these varied body types, according to Meyer, you need new genes. Those new genes have to encode new proteins that have new protein structures called protein folds. And if you look at the way new protein folds have been made in the lab, it's just too improbable for those folds to have occurred in geologic time, in the time that is required by the Cambrian explosion. So basically, you have improbabilities and too little time and so on and so forth. It's more complicated than that, but I think that's a fair distillation of his thesis. Now, all 3 of these planks are demonstrably false. Let me just put that right on the table, and that's why I disagree with the way he got there. So here's the Cambrian explosion. The Cambrian starts at about 543 million years ago. These are dates from radiometric dating. These are very well accepted dates in the geologic community. This division of the Cambrian, known as the Atmedanian, constitutes essentially the Cambrian explosion. And you can see there's not a lot of time there in the Atmedanian. It's about 530 to about 526 million years or so. Within this time, before that, you see what's so-called small shelly fauna. Very tiny little fragile little shells and things like that. You also see lots of trace fossils, sponges, things like that. It's not really until the Atmedanian that you start to see these remarkable organisms. Lots of hard parts, lots of jointed limbs and antennae and fangs and mouth parts and things like that. Remarkable diversity. So that's why it's called the Cambrian explosion. It's not because some nuclear bomb went off during that period. It's because you have an explosion of diverse life. Now, if you look at the number of species that come into the fossil record during the Cambrian, you'll notice something very curious. So this period that's called the Cambrian explosion is right here, yet the vast majority of diverse species are actually found after the Cambrian explosion. Secondly, something I'd like you to see, while you don't have a lot of diversity, you have plenty of creatures that come up to the Cambrian explosion. So I think one of the mistakes Meyer makes, he takes the Cambrian explosion and rips it out of its geologic context and says, look at this diversity. He pays virtually no attention to what comes before it, only to say that there's really nothing that can act as a precursor for what happened during the Cambrian explosion. Well, prior to the Cambrian explosion, you have this period of time known as the Vendian. And during the Vendian, you see sponges, you see some weirdos, but you see a lot of trace fossils. What are trace fossils? These are fossilized burrows and furrows and things that were left by the creatures that made them. The creatures that made them were not fossilized because they were soft-bodied and they were tiny. So tiny that they were the same sizes as the sand grains that would have fossilized them. That's why they're not preserved. This is sandstone. Sandstone has bigger grains than, say, shale or limestone or something like that. So that's why you see the burrows and the furrows, not the animals themselves. So scientists, paleontologists have named the burrows that were presumably made from a particular animal. Now, the first burrows are very, very early in the Vendian, somewhere around 570 or so million years ago. Those are very simple. They're just straight furrows like this through the bacterial mats. As time goes on, these burrows get more complicated. They start to branch. Instead of just going through the mats, they start to actually go down horizontally. You start to get things that are now curlicues and things like this. When you hit the Cambrian, you now start to see things like this, where animals are starting to go up. And this means that they're coming up to feed. That's an unusual behavior. Now, what does it take to make a burrow like this? You have to have a fluid-filled body. In order to have a fluid-filled body, you also have to have coordinated muscular movements of the body. That takes complex muscles. That takes a complex nervous system. That takes a complex brain that coordinates the movement of these body muscles. You also have to have a central cavity that gives you some hydrostatic resistance against what you're pushing against. Basically, you have to have a three-layered critter in order to do this. So the three layers from which our bodies develop, ectoderm, mesoderm, endoderm, were all present before the Cambrian explosion. This means that the genes involved with the formation of these three layers all had to be present before the Cambrian explosion. Now, if you look at the development of a variety of vertebrates and invertebrates, you see something amazing. You see the same sets of signal transduction pathways that are used in the development of starfish, sponges, jellyfish, fish, frogs, Fred, apes, whatever. The same pathways that use essentially the same genes. Why then do we get these different body types? It's a different way these pathways are deployed. It's a different way these pathways are deployed. So do you need new genes? No. You need to have novel uses for the genes you already have. And that is a big knock against Meyer's thesis. So what do we see when we go to the genomes of these relatively simple vertebrates? So first of all, if we look at sponges, sponges are relatively simple. We have a body cavity with feeding cells. We have essentially amoeba-like cells that digest things. I want you to look at these collar cells here. That's your digestive cell. It sweeps in material into the cell, that collar cell, and this is a protozoan here called Monocyga. Do you see a resemblance here? You should. If you look at the genomes of Monocyga and the genomes of the sponge, they're very similar. Very similar. In fact, Monocyga is the most similar unicellular organism to all the animals. So what happens if you look at Monocyga and then these other organisms? Something remarkable falls out. In these unicellular organisms, you see some of the pathways that are used in making multicellular embryos. Then, if you look at Monocyga, you see this pathway, but then you see the beginnings of these other pathways. Those pathways are there, but they're not quite fully formed. They'll have the transcription factors, but not the receptors. Or the receptors are not the transcription factors. They will have some of the machinery, but yet it's not all put together yet. If you go to Trichoplax, a flat little weirdo thing that only has 4 different cell types, now you start to see more of these various pathways. And then Amphimedon, a sponge, you see almost all of them, and the ones you don't have, you see the beginnings of them. When you go to coelenterids, things that like corals and jellyfish and things like that, now you see virtually everything, and even those that you don't see, you see the beginnings of that pathway there. Essentially, when you get to certain sponges, now you see all of these pathways here. This means, and by the way, these genes that you see here were probably not derived independently because these genes are so closely related to each other. They were probably inherited from a common ancestor. So what does this tell us? This tells us that the genetic toolkit required to make these different body types were available long before the Cambrian explosion. Secondly, new body types are generated by new deployments of already existing genes, not the creation of new genes. Thirdly, he talks about protein folds and the evolution of protein folds. What's a protein fold? A protein fold is a specific arrangement of a protein chain in 3D space. Certain protein folds are associated with certain functions. Example, Rossmann folds. Rossmann folds bind ATP. ATP is the energy currency in the cell for doing stuff. If you want to make something, you gotta pay for it. How do you pay for it? You use ATP. Their wallets are full of ATP. That's how you make stuff in cells. Rossmann folds are essentially the wallets of proteins. They bind ATP. That's just one example. Let's take a look at how you can make new protein folds in the laboratory. This comes from the University of Maryland. This is Philip Bryan and his colleagues. These are two different protein folds that come from a bacterial protein. You can interconvert those protein folds with one amino acid change within those proteins. You can change this one to this one. Then with another single amino acid change, you can change this back to this one. And these have different functions. One binds antibodies, one binds proteins that are found in the blood. Interconversion of a protein fold with one mutation. This is not a fluke. Researchers in Sweden have shown a similar thing with the stinging cells of jellyfishes. There's a protein in the wall there that has a support protein. These two proteins, it turns out their different protein folds can be interconverted with just one amino acid change. Furthermore, protein engineering experiments have shown that you can make novel protein folds through protein engineering. These are called beta barrels. Beta barrels act as channels in cells that let things in. Some very enterprising people took two proteins that were completely unrelated and were able to fuse them together and were able to make a beta barrel. Two unrelated proteins that didn't look anything like this, they were able to make a beta barrel. This is called an alpha-beta barrel. Alpha-beta barrels are found in lots of different types of proteins, enzymes in particular. This is a half-beta barrel. This is completely functional. Bridget Hooker in Germany has shown that you can take these half barrels, you can fuse them together to make these types of proteins, and now you get proteins that have these types of functions. She's also been able to do this with other types of proteins that have even smaller versions of this. That's called a ferredoxin fold. If she fuses the ferredoxin folds together, she gets proteins that look like this that are functional. And if she fuses these together, she gets proteins like this that are functional. And this is just a summary of Hawker's work. I won't go into that because interest of time. Also, there are these things called circular permutation, where you can duplicate genes and then give them haircuts. And you can produce completely novel arrangements of protein domains. If you do this, you have a new protein with a new structure with a new function. You can do this in the laboratory, and then you can trace how protein history, essentially, has been created through things like this. And then if you try to redo this in the lab, recapitulate these evolutionary steps, you get proteins that completely work. Nick Grisham at the University of Texas at Austin has done this. And there are protein families that very clearly have been made through circular permutation and other mechanisms like this. These are just a few of the many, many different types of examples of proteins whose folds are changed, whose structure are changed, whose functions are changed, and those proteins are completely stable and, in many cases, completely functional. So to summarize this, protein engineering has shown you can make new, stable, functional proteins from things like gene fusions and non-homologous recombination experiments. These protein families can arise by means of circular permutation or gene duplication or other sorts of mechanisms. Protein folds can be converted into new folds through even point mutations. None of these events are improbable. Meyer tries to argue that these events have the probability of 1 × 10⁻77. That comes from experiments from Douglas Axe. He misinterprets those experiments wildly. So let me just say, I believe in the designer. But trying to prove his existence with bogus science doesn't do anybody any good. And I quote from one of my personal heroes, Agustin Aurelius de Togaste, the bishop of Hippo, from his literal meaning of Genesis, Now it is a disgraceful and dangerous thing for an infidel to hear a Christian, presumably giving the meaning of Holy Scripture, talking nonsense on these topics. These topics happen to be astronomy in this case, but we could say biology. And we should take all means to prevent such an embarrassing situation in which people show up vast ignorance in a Christian and laugh it to scorn. Thank you.